Hi, this is Dr. Michelle O'Donoghue reporting from Medscape. I'm here at the American Heart Association meeting, and one of the interesting stories that we're going to be talking about is that of gene editing. So joining me to discuss this is Dr. Sekar Kathiresan. He's the co-founder and the chief executive officer of Verve, and they presented some very interesting results. So before we get into the results, let's just maybe take a step back and talk about the field in general. There are a lot of people who may not be familiar with gene editing, so maybe you can just walk us through a little bit of the basics.
Sure, Michelle. Thank you so much for having me. So gene editing, as you know, the technology was initially described in 2012 by Emmanuel Charpentier and Jennifer Doudna. And the first generation of this technology are so-called genetic scissors. That's really the analogy. And that's CRISPR-Cas9. And how CRISPR-Cas9 works is when given a guide RNA, it goes to a specific place in the genome based on that address that you gave in the guide, and then directs a cut. So it basically cuts the DNA entirely at that intended spot. And it's been used to basically turn off genes. That's really the major way that it's been used. The first of the medicines using that technology, called exa-cel, being developed for sickle cell disease, is the furthest along and likely will get FDA approval by December. That's the first generation technology. The second generation technology is called base editing. And base editing differs from standard CRISPR-Cas9 in that the base editor doesn't cut DNA, but rather chemically converts one letter to another. So it's more like a pencil and eraser, where it can go to a specific spot in the genome, erase a letter, let's say an A, and change it to a G. And that can be used to either fix mutations, or in our case, we're using it to turn off a gene as well.
There's a lot of potential here, certainly. And I know that PCSK9 is a target of particular interest for you. So that's where you're starting. And you want to maybe walk people through what you have right now and what you're presenting today?
Yeah, so what we presented today are results from the heart-1 trial of Verve 101. This is a first-in-human study of this new technology. Why are we doing this? You know, there are other medicines available right now to lower cholesterol, siRNA, monoclonal antibodies. The major issue that we see is that despite the availability of all these medications to lower cholesterol, the fraction of patients who are actually on an LDL-lowering medicine who've already suffered a heart attack is actually very low. We think a lot of that has to do with the chronic care model. The idea that somebody has to take a daily pill or intermittent injections for decades for LDL care. What we'd like to do is replace that chronic care model, really disrupt that model with one-and-done treatments. So imagine a future where you get a one-time intravenous infusion and a lifelong LDL lowering. That's the idea behind Verve.
We know that non-compliance is an important issue. And a lot of our patients, where we're sending them out the door with prescriptions for statins, for instance, a lot of those prescriptions are going unfilled. And even with the PCSK9 inhibitors, we now have some of these therapies that last longer, but certainly not the one-and-done approach that you're talking about. And now that we're seeing patients who seem to be having more and more early onset of premature coronary disease, the idea of facing a 40-year-old who needs to take a lifetime of regular treatments, it's very daunting for them. So I think that there is, certainly appeal in the idea of approaching this through gene editing. Now, what do you say, though, on the flip side about people who express concerns about this type of an approach? I mean, one and done is great on efficacy, but of course there's going to be the concerns about the safety ultimately.
There are kind of several aspects of safety, right? So one is the gene itself. What gene are we turning off, and why do we think it's safe to turn off the PCSK9 gene permanently? That actually comes from human genetics. So there are people walking around who have this gene naturally switched off lifelong. And they have very low LDL, and they're remarkably protected against heart attack, and they're otherwise healthy. So this is what gave us the inspiration that there are some genes in our body that are truly spare parts. You can get rid of them, and all you get is health. And PCSK9, believe it or not, seems to be one of those. And so our goal is to basically switch off the PCSK9 gene in the liver to mimic that natural situation where people are protected. And so we started with this idea in 2018. 2020, we got it to work in non-human primates, monkeys. And then last year, we dosed our first patient. And then this year, we've announced the results from the first 10 people in the phase 1 trial. So the drug itself is an mRNA for the editor and a guide RNA. Both of those are packaged in a lipid nanoparticle that lipid nanoparticle is infused into the bloodstream. So it's a one-time intravenous infusion. Very simple. It's a peripheral IV. After the infusion, the drug makes its way to the liver. It's taken up almost exclusively by liver cells. Once inside the liver cell, ultimately the editor goes to the right place in the genome, makes the A to G spelling change at one spot in the PCSK9 gene to switch it off. Switching off the gene reduces protein production. The blood PCSK9 should go down, the blood LDL should go down. That's the theory. So we've treated 10 patients so far. Now, what type of patients should we treat with this kind of new therapy, brand new modality, first of its kind? So the FDA has guided last year on the appropriate patients for a phase 1 clinical trial that incorporates human genome editing products. And what they said was that you should study people with severe advanced disease. As you know, most phase 1 studies are healthy volunteers, but the patients we've enrolled are by no means healthy volunteers. They're the sickest of the sick. The patients we enrolled have heterozygous FH. They already have established ASCVD, often multiple procedures, multiple myocardial infarctions. The mean LDL cholesterol was 193 on maximally tolerated oral therapy. So we had these patients, we enrolled them. There have been 4 dose levels that have been administered, 3 patients at each of the first 3 dose levels, 1 patient at the 4th dose level. The first 2 dose levels, there was minimal response on either the PCSK9 or the LDL after infusion. The 3rd and 4th dose levels, the potentially therapeutic doses, we've treated 3 patients. And what we found at the PCSK9 reductions were a range of 47% to 84% reduction. The 84% reduction is pretty sizable because about 85 to 90% of the blood PCSK9 protein comes from the liver. So the fact that in that one person we've got an 84% reduction means we probably edited nearly every hepatocyte. Both copies of the gene turned it off. In terms of LDL, the real clinical endpoint, we didn't see much of an effect at the first 2 dose levels. At the 2 highest doses, we've seen LDL reductions of 39%, 48%, and 55%. Range that is well within what the current medications available offer and within what our therapeutic product profile was. Now, let me move to durability. We have durability data out to 6 months right now in the longest treated patient with the efficacious dose. And the LDL came down 55% at 28 days, and then 6 months later, the LDL is still down 55%. Very reminiscent of the non-human primate data, the monkey data, where a one-time treatment, and then 2.5 years later, the LDL is still down 60%. So this really gives us some confidence that with longer follow-up, we'll have the durability that we're looking for, but that follow-up is ongoing.
It's in line with what we'd expect to see with the PCSK9 inhibitors that are currently available. But here, the durability piece is, I think, what's really intriguing and exciting.
Yeah.
And then what did you learn on the safety side, appreciating that it's small numbers?
Yeah, small numbers, and it's a phase 1 in patients with severe ASCVD. So we saw 3 things. The first 2 are LNP related, so the lipid nanoparticle infusion related. In patients at the 0.45 and 0.6, the 2 highest dose levels, patients experienced a flu-like symptoms during infusion and shortly thereafter that resolved pretty much on its own or sometimes with just Tylenol. These symptoms are fever, body aches, and headache. This is something that's expected. The second is there's a transient rise that's reversible in liver function tests, specifically ALT, with the infusion in the 2 highest dose levels. The peak ALT is 4 or 5 times normal, but then comes back down, and within a week or so, it's back to normal, no evidence of liver injury. Again, that was anticipated based on the preclinical data in non-human primates. And then of the 10 participants, 10 patients, 2 have had cardiovascular events. And really due to their underlying severe ASCVD. 1 patient suffered a fatal cardiac arrest 5 weeks after infusion. That patient had a history of prior cardiac arrest and ischemic cardiomyopathy at baseline. There was an autopsy done. The autopsy showed severe underlying coronary disease and no evidence for myocardial inflammation, myocardial infarction, or a pulmonary embolism. And so the DSMB determined that that event was unrelated to treatment. The second participant suffered a myocardial infarction, and that was determined to be possibly or potentially related to the treatment because of the proximity to the dosing. However, this patient had unstable angina coming into dosing, chest pain with exertion that he did not disclose to the investigators. After the treatment, he went to angiography, and the angiography showed multivessel coronary disease, in-stent restenosis involving all the major arteries, as well as an obstructive lesion in the left main. These were all pre-existing, and I think it was unfortunate the patient had not revealed the symptoms that he had. But as you know, these things happen sometimes in early clinical studies. And again, the DSMB felt this was due to the severe underlying disease and encouraged us to continue dosing. These data have also been reviewed by the FDA when we submitted our IND clearance request, and they've cleared the IND now, and we are now ready to initiate this study in the United States. Currently, the study is ongoing in UK and New Zealand, and now with the US clearance, we'll be able to start enrolling patients in the US.
As you say, these are sick patients, and they're small numbers, so ultimately we will need to see the safety profile in a larger group. But I think in terms of the efficacy, I think it's very interesting to see these early results, and the durability will be, terrific to watch as time goes on.
There are two major takeaways, I would say, from this work. On the scientific side, this is really the first time that it's been shown that you can change a single letter in a single organ in a human being for a clinical effect, so-called in vivo base editing in humans. On the clinical side, it's early, but this could open the door to an entire new way to treat heart disease. Rather than daily pills, intermittent injections, a one-time intravenous infusion procedure, and then lifelong LDL lowering.
So to that point, what else do you have in your pipeline? Because as you say, it opens the door to many different possibilities.
We've been very focused on the different axes of lipoprotein risk for heart disease. And as you know, cholesterol in the blood is carried in any of three different particles: LDL, triglyceride-rich lipoproteins, and a particle you know very well, lipoprotein(a). The PCSK9 program addresses LDL. We have another program that targets the ANGPTL3 gene that turns it off in the liver. That targets LDL and triglyceride-rich lipoproteins. And then we have another program that targets lipoprotein(a). So we envision a future where the appropriate patient may get either one or a combination of these treatments to get lifelong lowering of blood cholesterol and avoid heart attack.
We'll be staying tuned because I think that there's a lot of interesting developments that we'll be seeing on this front in the years to come. So thanks again for joining me today, Sek.
Thank you so much, Michelle.
Signing off from Medscape, this is Dr. Michelle O'Donoghue.